It's my pleasure to start off our afternoon by introducing Dr. Theoharis Theoharides. Dr. Theoharides is a highly esteemed professor and vice chair of clinical immunology. He currently serves as the director of the Institute for Neuro-Immune Medicine Clearwater and is an adjunct professor of immunology at Tufts School of Medicine. He earned his BA, MS, MPhil, PhD, and MD degrees from Yale University and was the recipient of the Winternitz Prize in Pathology. Additionally, he holds a certificate in global leadership from Tufts Fletcher School of Law and Diplomacy and completed a fellowship at Harvard Kennedy School of Government. Dr. Theoharides underwent internal medicine training at the New England Medical Center, where he was awarded the Oliver Smith Award for Excellence, Compassion, and Service. Dr. Theoharides is a leading expert on mast cells, having demonstrated the crucial role in neuroinflammation, particularly in the development of autism spectrum disorder, chronic fatigue syndrome, and long-term COVID-19. His expertise has earned him induction into the Alpha Omega Alpha National Medical Honor Society and Rare Disease Hall of Fame, which I didn't know there was a Rare Disease Hall of Fame. That's pretty impressive. And the World Academy of Sciences. Dr. Theoharides is a prolific inventor, having developed novel dietary supplements and a part of the Institute team. So we welcome Dr. Theoharides.
Okay, so I'm delighted to be here. I'm delighted to be able to talk about complex problems. I'm delighted to be part of the Institute. And hold on to your seats because you're going to hear so much science that you're probably going to get tired of me for the next 30 minutes. But it is this kind of science that I hope will allow us to explain many of the clinical information you've already heard and probably more to come. So everything that I'll talk about can be found either in whatever the Institute will actually put on site, as well as on the website, in the first one, which is the easiest to look at, which is drtheoharides.com. I urge you, if you have the chance, either directly or through the pod site of the Institute, to look at this YouTube presentation, My Mystery Symptoms and Mast Cells, because many of the things I'll talk about are presented through two patients that might find very close to your heart in many ways. So there are a number of things that you've already heard about, and I will try to kind of clarify some of those, and then we'll go into the science. First, you already know that especially MCAS, it's a complex disease. It's kind of different presentations. But I will stress as we go forward what is presented actually in that paper, where they talk about brain fog. And we will talk about brain fog, what that means, because it can be explained in many different ways. And there's a little caricature there as to what brain fog may be and how you might get to it. But very important is that many patients with MCAS present with numerous other conditions, such as fibromyalgia, such as ADS. Of course, we'll be hearing about Gulf War illness, etc. So the question is, are these comorbidities, or are they actually different presentations of the same underlying pathophysiology, if you wish? And we don't know the answers of yet, but many of the findings, as I will describe, seem to indicate that there might be a common denominator to all of those. So, for instance, this wonderful paper was published recently with many, many, many colleagues being involved. There were too many colleagues to actually include the co-authors. And there are 2 points I want to make, even though it's such a complicated story. First of all, they call it deep phenotyping, which is what Nancy has been doing with her colleagues here as well, is to find out characteristics of these patients that might allow us to either identify them or separate them from other groups. And they collected patients from all over the United States. So at the end of the day, this caricature, which is not really very informative, but it does tell us that there might be issues with the periphery, meaning outside the brain. There might be issues with the nervous system. We call it afferent messages getting to the brain. There might be issues within the brain. There might be issues with neurons sending messages outside the brain. Again, we're talking about periphery and then the outcome. So all in all, we're talking about everything is possible. Okay. However, I actually used this red box, which moved a little bit, to indicate that a lot of things will be happening in a part of the brain that is called hypothalamus. Now, why? Because hypothalamus is the master part of the brain that regulates what we call homeostasis. And what is homeostasis? It's the ability of the body to balance all actions in the body so that we survive and live happily thereafter. But if things happen to the hypothalamus, a lot of things can go eerie. The hypothalamus talks to the pineal gland that regulates whether we sleep or we don't sleep. It talks actually to the hypophysis or pituitary that puts out all the hormones that regulate our body, et cetera, et cetera. So when we talk about inflammation, as was part of my title, that's in a way a misnomer, because inflammation is very different in different parts of the body. If we have eczema, it's inflammation in the skin. If you've got asthma, it's inflammation in the lungs. If you've got multiple sclerosis, it's inflammation in your brain. But all inflammation is not the same. So we should try to avoid general terms that don't quite fit what we're trying actually to do. The other part that is very important about this study is that they looked at the expression of genes. We have 35,000 genes. And some genes get activated, some are dormant, some might be activated after we're exposed to something, might be environmental, viral, et cetera. And they found that a number of genes in both males and females were activated. So as you go upwards towards the right, that means you're activated. And they were actually associated with the immune system. So something is happening with these MCAS patients. But again, the immune system is general. We don't really know exactly what is happening. The second point is that these genes were slightly different in females than males, which might explain why things might be happening more often in females than not. This information doesn't tell us what to do yet, but I will base a lot of what I will tell you in a few minutes on the fact that it fits into this overall phenotype. So where do we stand? So we said some years back, and we continue to say that, and many of my colleagues here believe that there might be both environmental and sort of neuroimmune triggers that might regulate what happens or dysregulate what happens in the hypothalamus. So if I were to actually get a piece of this hypothalamus here, and this is a little caricature of what we call the blood-brain barrier. So the brain is protected by a set of cells that makes it a little difficult for things to get from the lumen inside the brain. So if the blood-brain barrier is disrupted, then it becomes leaky, and things from the blood can get into the brain, and the brain will freak out because they shouldn't be there. And the brain does not have circulating immune cells. It only has the so-called microglia. So think of the microglia as spiders making spider web, and the neurons crawl on the spider web, which is scaffolding, and make connections. So if the microglia get activated, they will stop making the scaffolding, and they release molecules that break down the scaffolding. So connectivity now is gone, and will release inflammatory molecules, causing a local inflammation. So if it is a localized inflammation in the hypothalamus, most of the time we're not going to pick it up in the blood. But in fact, many times we're not be able to pick it up in the cerebrospinal fluid either. So that's why I hate the word inflammation in general, because they would say, well, why do we measure cytokine X in the blood? And it's negative. It doesn't mean very much to me because something is happening in that localized part of the brain. And here you can see a number of papers talking about the possibility of focal or localized inflammation in the hypothalamus. And as we heard earlier today, the effect of stress. I can give you 3 hours lecture on just stress, but this is to indicate that certain cell type that we will be talking about can in fact be affected by stress. So stress, it's not only your mind going to shrink, something is really happening. And very recently, there were papers written about exploring joint potential, which is what we're talking about, of inflammation, immunity, and receptor-based biomarkers. This is just jargon to say that a lot of different things can converge, basically, to allow us to potentially understand what may be happening. And I will stress quite a bit in a few minutes about what we call mast cells. What are the mast cells? So an experiment was done back in 1901 where two scientists, Portier and Richet, were doing a simple experiment in the Oceanographic Museum of Monaco. They were going to take tentacles from jellyfish, because there are a lot of men of war in the area, inject them into dogs, and they were pretty sure the dogs were going to survive any subsequent stings. And they had a word ready for it. The word was going to be prophylaxis. When they did the experiment, the dogs actually died, so they called it anaphylaxis, which was the opposite. At about the same time, Dr. Paul Ehrlich, who got the Nobel Prize a few years after that, was staining tissues with various dyes. And he was very surprised because he used a dye that was blue, and certain cells turned out violet. And some cells were very compact, some cells were a little kind of spread out, so he could see those dots, but he didn't know what those dots were. He thought that these cells were feeding other cells in the area, and from the Greek word breast, such as tectomy, for instance, etc., they called them mast cells because he thought they were feeding surrounding cells. He was wrong, but he got the Nobel Prize anyhow. He was a wonderful scientist, regardless. Now, where are those cells found? If we look at this picture, all the red arrows indicate these violet cells from our laboratory. All of them are found perivascularly, very close to blood vessels. And we heard earlier about neurovascular problems, and that's exactly what I will indicate in a few minutes. They come from the bone marrow. They're found everywhere in the body. And I and other colleagues believe they're extremely important because they're the first cell to get activated when they see danger. So they're found in our eyes, nose, mouth, lungs, gut, skin, so they're exposed to the outside world. But they also throw tentacles through the endothelial cells that make up the walls of the blood vessels, and they sense what is happening in the lumen. So they will pick up danger immediately within seconds, as you will see. And you might ask, how do they actually sense what is happening in the lumen? This was a wonderful paper where they show a blood vessel, a mast cell sitting in the bifurcation of the blood vessel, and the arrowhead, which is difficult to see really, because this was a video, show that the mast cell throws, as I said, filopodia. They sense what is happening in the urine, and they will get activated from things that we never expected. And even though I will not have time to tell you in detail, in the journal Science, about a year ago, excellent journal, colleagues from England published that the mast cells that we thought mature only about 2 years after a child is born are actually activated in utero by whatever the mother is exposed to. It's a whole different story that might lead us to what might be happening in autism, for instance. So this caricature is everything you ever want to know about mast cells. So we teach students that they're activated by immunoglobulin E, which sits on specific surface receptors. And when it's bridged by antigen, it leads to a flood of calcium ions, which within seconds leads to release of molecules such as the well-known histamine, but all other molecules such as platelet activating factor, tumor necrosis factor, and meat tenderizers like tryptase. And if that happens, that's what the mast cell looks like from our laboratory. This is the nucleus. The dense granules are still intact, and the rest of the granules now have released their content. So think of it as a soccer ball filled with about 1,000 ping pong balls, each ping pong ball containing about 50 marbles. So if you're unlucky, you're stung by a wasp and you go into anaphylactic shock, this damn thing will degranulate, will release about 100 different molecules, of which histamine is only one. In 24 hours, and the laboratory is ready to fire again, so it doesn't die. So you constantly have an army waiting for something to happen. And unfortunately, since 1947, when histamine was discovered inside mast cells, we only have antihistamines, which block the action of the histamine after it's been released. So we don't have a good way to block the mast cell from releasing its 100 different mediators. However, since about 7 years ago, a new receptor was discovered, which responds to various drugs and other substances that are positively charged. We call them cationic. And that has changed our thinking about what mast cells can do. And in fact, this receptor seems to be talking to the other receptor, which might explain why some patients that might be allergic to something get activated by something else. Now the mast cells are ready to fire to everything you might imagine. This is all we teach, if in fact we teach all of this to medical school. The rest of it, we don't, for reasons that I don't understand, which is the mast cell, and of course, these receptors are all over the surface. I just indicate pictorially where they are. We have receptors for molecules released from nerves. We call them neuropeptides. Receptors for molecules released for other immune cells. We call them cytokines. and receptors called TLRs, or toll-like receptors, that fire in response to bacteria, fungi, and viruses. And in fact, this caricature and this caricature right here were taken from a review my colleagues and I wrote in the New England Journal of Medicine back almost 10 years ago. And it indicates a mast cell in the middle that can be triggered by so many different molecules that have nothing to do with allergy. And what happens when the mast cells are activated? Within 6 and 24 hours, they release molecules. The first line is cytokines, the second is chemokines, the other is peptides. They're all pro-inflammatory, but the mast cell does not degranulate and does not release histamine. So in fact, by looking at degranulation and histamine release, we're missing the forest for the tree. And it is this that I believe is happening in the hypothalamus in conditions like the ones we're discussing. So can I show you, in fact, that this indeed happens? These are 3 papers published from our laboratory in the Proceedings of the National Academy of Sciences, all 3 of them. In the first one, we stimulated the mast cells with a neuropeptide coming from nerves called substance P, together with a cytokine release from other activated immune cells called interleukin-33. And we measured release of tumor necrosis factor, an extremely potent pro-inflammatory molecule. You get thousands of units released. Control is without stimulation. We did the same thing. We measured interleukin-1, which is another very important pro-inflammatory molecule. Again, hundreds of thousands of units with nothing in the control. And then we did the same thing. We measured a molecule of vascular endothelial growth factor that makes the blood vessels leaky, both in the periphery and in the blood. And as you can see, as we increase the concentration, we get very high amounts of VEGF. Taking these 3 together, the mast cell can be more important than any other cell in the immune system, releasing pro-inflammatory molecules and molecules making the blood vessels leaky, as I said, both in the periphery and the brain. And now stress, we heard about stress. Well, why does stress affect anything? Well, here, human cultured mast cells stained with an antibody that recognizes on the surface the receptors to which the first hormone released under stress binds. It's called corticotropin releasing hormone. And the mast cells light up like light bulbs. So the mast cells can, in fact, be stimulated by a stress hormone released in the brain and outside the brain. There's no question about it. So if someone is telling you stress is making my eczema or whatever worse, go and get an antidepressant, they're crazy. It's happening, and we just don't know how to deal with it as of yet. And to make things even worse, when peptides like neurotensin or substance P stimulate the mast cell, they also make the mast cells produce more receptors for the stress hormone. So now the stress hormone activates the mast cells even more, and you keep basically a process going. So we need to be able to inhibit the mast cells, and hopefully for the diseases we're dealing with, in the brain, which means we have to come up with something that gets in the brain, which I will tell you about shortly. But as I said, the mast cells respond to pathogens. So here a caricature showing a mast cell talking to macrophages, talking to T cells, which are very important for parasites and viruses, and they can be activated by these TLR receptors. And I just chose a few papers here to indicate, as I will show you with the next slide, that mold is an extremely important trigger of mast cells and the microglia in the brain. And here, for instance, you can see sporothrix, which is one type of mold, can stimulate the mast cells to release cytokines, interleukin-6 and TNF, with no degranulation. And we've written some, and so have others, about the importance of mycotoxins in brain pathophysiology. Well, my colleagues here have already published a paper showing that certain types of mold can release actually mycotoxins, especially ochratoxin, in the urine of patients with MCAS. There are papers now talking about the neurotoxicity of ochratoxin A, and we recently published a paper where we added ochratoxin A to the so-called, as I said, microglia, the defenders of the brain, and we measured release of either interleukin-1 or interleukin-18 or a chemokine. And the last bar here is actually ochratoxin compared to other molecules, and the ochratoxin can stimulate the microglia. So therefore, environmental triggers could actually do a job in the brain defenders, and we will not be able to see that because we cannot see this visually in any way. And going back to this publication from the New England Journal of Medicine, I want to indicate that when the mast cells are triggered, depending on the trigger, they can release different molecules and can affect every organ or tissue in the body. Therefore, many of these patients show up and they have 20 different symptoms, and every organ in the body is affected. And many of our colleagues say, oh, you're crazy. You can't have all of that. And in fact, they do have all of that. And to make things even more complicated, we separate mast cell disorders in primary, which is what we call mastocytosis. If you have too many mast cells, and especially in the skin, you can see it. Or secondary, which is the bread and butter of what we see, allergies, rhinitis, eczema, etc. And then anything medicine doesn't understand, we call it idiopathic. Okay? So we do have now what is popularized as mast cell activation syndrome. And a lot of people keep on talking about having that. But without going into details, I need to tell you that diagnostic codes exist not only for mast cell activation syndrome, but mast cell activation unspecified, where you don't fit all the criteria for syndrome, and therefore you don't have to actually go and say, well, do I have MCAS? No, you can have mast cell activation regardless, and MCAS might be just a subclass of that. And we also use confusing definitions such as allergic diseases, atopic diseases, if primarily IgE is involved, or mast cell diseases or disorders or mast cell activation disorders. So are making our life difficult and even more yours, so to speak. So what happens if you do have mast cell activation? Could anything be happening in your brain? Well, here are some publications from different colleagues talking about neuropsychological and neuropsychiatric fixtures in mastocytosis patients, and a recent paper where they used mast cell deficient mice, and the mast cell deficient mice actually had much better cognition, meaning finding their way through a maze, et cetera, et cetera. So what about the mast cells of the brain? I mean, I did tell you they exist there, but do they really? So I published this review back in 1990 where I called the mast cells the immune gate to the brain. Why? Because if you see a caricature of the blood-brain barrier, you see the endothelial cell in violet, the pericyte sort of in blue, and a mast cell can hug that. Well, that's a caricature. Does it really happen? So here is actually an electromicrograph from our laboratory, 30,000 times magnification. You see the lumen of a blood vessel in the brain, a red blood cell in the middle. E is the endothelial cell, P is the pericyte. And here's a mast cell hugging basically on the blood-brain barrier. And what does it exist? It's found basically in the meninges, the membranes that cover the brain, as well as in the hypothalamus and amygdala. As I said, hypothalamus regulates homeostasis and amygdala regulate emotions. And we published, here's one of many papers, that if the stress hormone is released, it activates the mast cells, it makes the blood-brain barrier leaky within seconds. So, is there any evidence that, in fact, the mast cells are close to neurons? Here are two different ways that the mast cells communicate with neurons. One, you can see the vertical line is a blood vessel from the brain. The wiggly here line is a nerve, and the mast cell is piggybacking like on a nerve ending. This is 300,000 times diameter magnified from electromicrograph from our lab. You can see part of one mast cell. You see the granules, another mast cell, the granules, and this is the end of a nerve. And the tiny little arrows indicate the vesicles that release neurotransmitters through which we communicate. Now think, half of each granule is this meat tenderizer tryptase. Well, just imagine what it will do to the nerve ending if it's released, and so many other molecules that are released. And the mast cells have different ways of communicating with neurons, not only by being in close proximity as here, but here we show by culturing human mast cells with human neurons that the mast cells put out philopodia. These are nanotubules through which they can actually release molecules that reach distant sites. So there's no question that the mast cells do communicate with neurons. But how the microglia? I said the microglia are the defenders of the brain. Here are papers supporting this caricature that the mast cells can talk to the microglia and back, but microglia and mast cells being involved in inflammation. And here are papers, not from us, that microglia can be activated by mediators released from mast cells, etc. So there is a dynamic process going on, and the mast cells are not just static. They respond to the environment. As I said, they can increase their firing, etc. And we actually wrote a review about a year ago or so when we looked at the activation of genes in both ME/CSF as well as COVID patients, and we heard a little bit about the possibility that they might have a common underpinnings. And as you can see from this publication, 8 genes were common between the two diseases, only 8, and all of these genes had to do actually with inflammation, the TLR receptors, and T cells that are involved actually in virus and parasites. So yes, there is very good indication that there is actually some common ground between the two. Now, how about COVID and long COVID? I'm indicating a number of papers, but I'll actually specify 4. One is a recent paper showing that the blood-brain barrier is disrupted in individuals with long COVID. I've been telling you about the blood-brain barrier already. Here's a paper that histamine potentiates the ability of the coronavirus to get into the endothelial cells that make up the wall of the blood vessels, hence neurovascular problems we heard earlier. And mast cells promote viral entry by formation of chymase spike protein complex. Chymase is another meat tenderizer like tryptase that is found only in the mast cells. So here is direct evidence indicating that the two molecules can get involved. And in addition, we recently published a paper also that platelet activating factor, which is released from mast cells and can cause clotting and microemboli, is also released from endothelial cells. And in fact, the spike protein can stimulate the endothelial cells to release puff from mast cells. So therefore, a lot of possibilities exist that are testable and could actually end up being potential surrogates for us moving forward. We decided to bite the bullet and see, does any of what I told you actually happens? So we published about a year ago in the New England Journal of Medicine a paper where we stimulated human microglia with either substance P or interleukin-33 or the spike protein. And we did the same thing on mast cells as well. And you see that the spike protein alone can make the mast cells release very high amounts of interleukin-1 beta. Then we said, well, how does that happen, though? Does it happen through the receptor that the spike protein binds to, which is called the ACE2 receptor, or some other way? So we pre-treated basically the mast cells in another recent publication with antibodies that block the various receptors. And as you can see, only when we block TLR4, we actually inhibit the release of interleukin-1 beta. Now, that's very important because the ACE2 receptor is important for the viral to get inside the cells and multiply. But here we're showing that an entirely different receptor is involved in inflammation, and it is inflammation that we see in long COVID patients, as well as the patients that die from COVID. And a recent paper indicated that, in fact, as we showed, spike protein can induce the TLR4, same thing we showed here, and it causes basically now cognitive dysfunction in mice, taking us back to the brain. Then we decided to do the same thing in microglia. So we stimulated the microglia with recombinant spike protein, and we measure various molecules such as interleukin-1 beta, a chemokine, etc. Make a long story short, when we blocked only TLR4, we inhibited the release. So both in the mast cells and the microglia, the well-known ACE2 receptor had nothing to do with inflammation. It was a different receptor, and therefore, again, we're barking up the wrong tree by creating basically approaches to inhibit only the ACE2 receptor. And to make things even worse, you can see here that when the TLR is activated, the TLR4, somehow it makes the target cells grow more receptors for the ACE2, so now that the virus can actually invade the cells as well. So again, it's a very cyclic kind of process. So coming to the end, is there anything we can do about any of this? So we've been studying molecules that exist in nature called flavonoids, And for over 30 years worth of work, one particular flavonoid that is called luteolin has actually risen to the top in terms of publications. And you can see antioxidant, anti-inflammatory effects, and recent reviews about being also neuroprotective. So you might ask, okay, so that's a flavonoid, it's a natural molecule. How about any drugs? Well, the only drug that is known to potentially be a mast cell inhibitor is something called cromolyn. So we decided to actually test cromolyn against luteolin head-to-head. So we pre-treated human cultured mast cells with the same amount of either cromolyn or luteolin for the exact same pre-incubation time, 10 minutes. And then we measured different molecules coming out of the mast cells. Control is flat. The actual activation is way high. And you see luteolin, and then you see cromolyn. So if you compare these two bars everywhere, luteolin was better than cromolyn everywhere. So why are we not using luteolin? Because it's not a drug. And physicians don't want to write supplements. And of course, many supplements are not pure. So yes, there are a lot of questions about them. But just keep in mind, there's very solid evidence that certain natural molecules, such as flavonoids, can do the job. So can we prove that this is the case? So we pre-treated either human culture mast cells again, and we pre-treated them. In this case, we treated them with substance P, with either cromolyn or luteolin or a new molecule called methoxyluteolin. And you can see we inhibit them nicely. And we pre-treated microglial cells as we increase the amount of the flavonoid, the pre-treatment. You can see the amount of interleukin-1 being released went way down whether we use luteolin or methoxyluteolin. And a paper was just published about a year ago showing that luteolin inhibits, in fact, the spike protein causing respiratory problems. So it's not just us saying so. There's mounting evidence that this is the case. So how can we show that this has any relevance, let's say, to MCAS? So we did a study where we induced basically in mice what might be equivalent to MCAS. So unfortunately for the mice, we injected them with poly-IC, which mimics a viral infection, and we put them into cold water to swim. So that the stress of the cold water, they had to actually work to stay afloat, and they had a viral. I mean, you can't go wrong with that. So we actually measured what happens to various molecules, and I'll show you only two, in this case, tumor necrosis factor and interleukin-6 pro-inflammatory molecules. When we gave them actually regular diet, I call them low diet, or actually high flavonoid diets containing luteolin. So you see how high the molecules were in the non-flavonoid diet, and then they kind of absolutely disappeared with the flavonoid diet. So we can actually show that at least in animals, the fatigue can be reduced. And since then, two very important papers were published in the last couple of years where they gave actually flavonols or flavonoids and flavonols are very similar in terms of the structure in normal individuals. So in this case, it was normal, healthy adults, but both the cortical or the brain oxygenation got better, as well as cognition was improved. And this was done in New York in men and women. And again, it showed that their cognition was increased. These individuals did not have a disease. They just managed to multitask and do things actually better. So 2 slides left. I helped actually a company called Algonaut develop a number of dietary supplements, but I want to show you 3 points with this slide and I'll be done. Number one, that there is now evidence, in addition to what I showed you, that, luteolin can actually inhibit both the mast cells as well as the microglia. So you see this green line showing where it inhibits, and therefore it will inhibit various pro-inflammatory molecules from being released. But we all know that in the United States, FDA cannot regulate supplements, as in other countries as well. If, however, you voluntarily want to ask the FDA to check for export, and only for export, then they check whether the company is FDA registered, which Algonaut it is, whether the molecules are made in good manufacturing practice facility, which they are, and if in fact they're assayed by an independent source, in this case Eurofins, for quality and amount. And then they give you this certificate of free sale. It says US FDA, and it's renewable every 2 years. So I'm just urging you, if you're going to use supplements, try to get pharmaceutical grade. Try to get them as pure as they can be, know the source, and the purity, because there can be so many interactions with drugs, especially in individuals that don't have a good metabolic system, that you might end up having problems. So when something says immune booster, don't believe it necessarily, because if in fact someone is MCSF, I don't want to boost the immune system. I want to actually inhibit it to some extent. So the words can be tricky because they're market employees, but they don't essentially say what we wanted to say. And for better or worse, I was given a patent about the use of these flavonoids for brain inflammation. So final slide, or next to final slide. What are the findings? So we believe that there is a combination of innate and external triggers that activate brain mast cells and microglia, especially in the hypothalamus. This release of pro-inflammatory and neurotoxic molecules can disrupt neuronal connectivity and make things, go eerie. What can we do? Obviously, if you know the triggers, especially mold, avoid them. And the mold releases volatile mycotoxins, as I said, like ochratoxin. Therefore, it's not just a measure of looking for black mold on your tiles. These are secret molecules. You can only measure them in the urine or antibodies in the blood, again, the mycotoxins. You can support brain health by giving calcium folinate. Calcium folinate is different than folic acid. In fact, calcium folinate is how it appears in nature. Companies have made it folic acid because it stays for a long time on the shelf. The reason being, folic acid has to bind to receptors on the surface of cells to get inside, and that gets activated by an enzyme called MTHFR to make active folic acid. Luckily, calcium folinate bypasses the receptors and can go in, and it's like high octane gas for the enzyme, so it works better. And there are certain cases, unfortunately, where we have what is called global folate deficiency, meaning there are antibodies against the receptor, and they block basically the lock, and the kid just doesn't get in. And if we measure that and they're high, then we might have to do exchange transfusions, et cetera, to get rid of them. And finally, what are we trying to do right now? Because all of this has to actually be put together. No single cell type can help us understand what is happening in the brain, especially in complex diseases. So what we're trying to do is create what is called a brain on a chip. So it's an instrument where we can actually put neurons in the middle, put actually on satellite wells, microglia, mast cells, endothelial cells, to mimic as much as possible what might be happening in the brain, stimulate these satellite wells separately, and then measure the health of the neurons so that we can get a baseline, and then we can use that to start pre-treating with natural molecules or drugs that might help us actually undo these conditions. So this is sort of a complicated, if you wish, set of instruments, but that's the only way to bring all the data I've been telling you together. And at the end of the day, the very last slide, nothing can happen without support from you, because many of the things I've been telling you, many of the things you've heard earlier, are not necessarily easily funded by federal sources because they're a little bit outside the box, if not way outside the box. So unless we actually get the funding to do what I tried to convince you at least makes sense scientifically to move forward, it's not going to happen. God bless you all, and thank you for listening.